So just to introduce Maria, she is CEO and founder of Lifebit AI. Maria is a thought leader and biotech innovator, expert in AI-driven drug discovery, biomedical information, and federated computing. She is also a passionate entrepreneur and has founded 2 companies, Innovation Forum Barcelona and Techstars-backed Lifebit. Prior to Lifebit, she was a biomedical researcher working on developing tools and methods that facilitate the analysis of big biomedical data and promote personalized medicine discoveries. This includes the industry standard programming framework NetFlow that has revolutionized the computational analysis of genomic data. Maria is a frequent industrial speaker and has spoken in many international conferences on the subjects of genomic workflows, the computational challenges of personalized medicine, AI, cloud, and HPC genomics and drug discovery, women in leadership, entrepreneurship, science ventures, among many other topics. So today, Maria will be discussing real-time disease surveillance using artificial intelligence and Lifebit REAL. And I will hand over to you now. So I'll just make you presenter, sorry.
Thank you, Mark, and it's a pleasure to be here.
Okay, and I can just remind everybody to type any questions into the chat box and we'll get back to those at the end. So over to you, Maria.
Perfect. So I hope everyone can see my screen. Thank you, everyone. So a little bit about Lifebit. So Lifebit is a leading software in the space for software solutions in the space of population genomics and precision medicine. We do not provide, we're not a point solution provider. We provide our clients with an end-to-end capability, ranging from the ability to easily discover and access patient data for different diseases, as well as real-world information that can be used for disease tracking and discovery, all the way down to all of the suite and toolset that you need to identify cohorts, properly manage the data, perform different analytics, and as well as get clinical decision support for improvement of clinical trials and patient diagnosis. And from all of that application set down to like transformative services that we provide across data infrastructure science and other topics to our clients. So today we'll be going to be focusing exactly, as Mark said, on Lifebit REAL, which is a disease tracking and discovery system that is based on knowledge graphs and deep learning pretty much algorithms. So let me start with the first part of Lifebit REAL, which is the disease surveillance. So first of all, why do we care about understanding disease prevalence and surveilling diseases? So there are 2 main reasons for this. First of all, our clients, which are top pharma as well as different biotechnology companies, they really need to be able to understand what is the disease prevalence in order to stay ahead of the competition, but at the same time also be able to optimize the R&D pipelines and pretty much focus their strategic investments, both short-term and long-term. So particularly how Lifebit REAL works. So we use all of the data that is publicly available across the internet, as well as AI, and we combine that with the proprietary Lifebit real-world evidence technology, as well as our NLP graphs. And we are collaborating, as I said, with many different companies, but especially with like BI, we're doing a really big project around this area of disease surveillance. So how do we go about this? So we follow 3 main steps, as you would expect. First of all, we ingest data from millions of articles around the web. Then we extract the relevant information. And then eventually we combine this relevant information into a reporting dashboard that is able to deliver the information in a form that can actually be consumed by users of all levels without the need, as Ben was stressing, to have hand-holding from an IT or expert engineering and data science team. So in the first part, what do we ingest and how do we do that? So we ingest millions of articles and entries from, first of all, scientific sources, but that's not enough, especially when you are trying to do, when you are trying to assess disease prevalence. We include, beyond the scientific sources, public health organization information, or then information coming from different expert organizations, news, obviously, outlets, and social media. And then all of that is pretty much ingested and then fed to our AI algorithm and then processed. Then, once we've have been able to identify across the web all of the relevant entries, as I said, from all of these different sources, and actually manage to ingest what seems to have relevance to our diseases, then the extraction of the information comes. And as you can see here, this is the process by where we actually take completely unstructured information and we turn it into structured information following certain rules that our NLP technology is using in order on how to actually model, structure, and then model this information. Finally, as we say, we combine all of that information into a report that can be delivered to users in a way that is easy for them to understand the information, number one, but more importantly, be able to work with that information and base decisions on that information. And here we will normally, in that reporting, will show pretty much the prevalence of a particular disease, and this can be infectious diseases, but as well as other general disorders across the globe, with information around how the disease seems to be spreading or what's the prevalence, how many people or animals in this case are being affected. We span from both humans and animals as well. And then we actually even see transboundary events, as well as pretty much infections that move from animals to humans. And then, as I said, all of this information, it is presented in an intuitive dashboard where the users can actually very easily consume it and click around and verify that information, because again, as Ben stressed just prior to my talk, it is very important that the systems are not black boxes, because the users need to be able to verify every information that is coming and be able to track back onto what was the logic, what were the sources even of the data that contributed to this pretty much prediction, and even what was the logic on the confidence levels that we assigned to that prediction. So of course, once you have identified the relevant diseases, the next question is, what are the drugs out there that can be used in order to obviously to cure those diseases? And so therefore, drug identification is very important. So the question again is like, why do our clients and our users care about drug identification? So the biggest deal of 2020 was actually AstraZeneca spending more than 6 billion to actually purchase an antibody drug conjugate for lung and breast cancer. And this drug wasn't even in the market yet, was in the clinical trial phases. As such, the ability to define a disease of interest and its prevalence and how relevant of a strategic importance that is to a big pharma or a big biotech, and at the same time being able to identify the different drugs that are either in development or can be developed, are essential for those companies to move into decisions around either in-licensing those drugs or optimizing their R&D pipelines in order to start focusing more the development around those drugs. So again, how does it work here? The same 3 steps. We ingest a lot of data from the scientific literature this time. Then we extract the relevant information, and then we combine it into a report that it is easily consumable by users of all types, as we said before, and it is verifiable by them, and it's presented in a way where decisions can actually be based on that information. So here I'm going to walk you through 3 use cases. So first of all, around psoriasis actually. So what the system is able to do here is to identify clinical and preclinical drugs. And here you can see that we were identifying drugs for psoriasis, and the system was able to actually identify the drug, the company that is manufacturing it, and what is the stage of the drug, but at the same time was able to infer and discover alternative indications and sometimes even novel indications. As you can see here with psoriasis, seems to also be having as an alternative indication, seem to also be working for autoimmune diseases, which, sorry, not the methotrexate here, which was identifying one of the drugs, was also identified that it is a drug that can be used for autoimmune diseases because it inhibits the immune system, so that was making sense. But also for COVID, as there were also some early work and publications that was actually suggesting that pretty much it can actually inhibit the virus spread. The second use case obviously is what we want to identify when we actually propose, identify the different drugs that are in the market or are in development or are novel and could be developed. Then we also care a lot about identifying adverse effects of those drugs. And one of the best ways that is not that much used, but it will be used in the years especially to come, is actually using social media articles and basically all of that information in real time to actually point out, like in this case, that methotrexate seems to be actually causing nausea and foggy headed and hair thin, and these are some side effects that are being reported by the users, pretty much creating what we call an experiential insight to how side effects of the different drugs can manifest during the life cycle of a drug being administered to different patients with obviously different genetic backgrounds. The third use case is obviously to try to now not just identify drugs that are in development or being developed, and their side effects on the patients that they are already administered or the patients that are receiving those drugs in the clinical trials, but also be able to identify entirely novel indications and target drugs. And here we, again, we are using a combination between our NLP technology, but also our deep learning technology, where from the different articles and the knowledge graph that is based on that learning, we are able to then very quickly understand the mode of action of these drugs, understand the pathways, and then pretty much suggest different genes, their indication, the clinical, the chemical that can be used, sorry, the findings, the genes, the indication, the chemicals, the genes, and the regulatory, the regulation pretty much networks that they seem to be regulating, as well as the different references into that. So you can always trace back from where this information is coming. Eventually, all of this information, as Ben was showing before, it is combined in a big knowledge graph. But this knowledge graph has information about not just which drugs can be used for which diseases, but actually and which drugs can be used for which diseases in a particular timing point, which is one of the major differentiators of our technology compared to other technologies. Our knowledge graphs are updated real time and take into account real-time information around pretty much the disease prevalence and all of the different drugs that can be used in a given time for that particular disease. And of course, then those knowledge graphs will also include further information around the mode of action of the drugs, the in-licensing opportunities, the novel use of those drugs, and so on and so forth. Then, of course, when you define relevant diseases, apart from needing to identify drugs that can be used for these diseases, a lot of times there is a need to develop vaccines and adjuvants, like in the case of COVID. So this technology is not based on NLP, is solely based on deep learning networks and ensemble networks around that, where pretty much we are able to do immune response profiling for vaccines and adjuvants. And then obviously the inputs that the system expects would be all types of genomics, proteomics, transcriptomics data, coming from different cell lines, animal models, and humans from different clinical trials. And obviously the output that the system is able to predict is the innate immune response in terms of like the macrophage response, fever, and cytokine profile, the adaptive immune response in terms of B and T cell response, antibodies and cytokine response more, and fever, and of course is also able to start suggesting novel adjuvant formulations. And this has been used so far. We've been using this so far across our clients in 3 cases. The one is to actually make predictions between in vitro and in vivo, temporal predictions where we are predicting on kind of like day 1, let's say, or from a particular time point that we have the data, what will happen 10 days later or a month later or 3 months later, as well as we said before, the ability to actually rank different adjuvants and antigens, so we can actually guide the optimization and the selection of which are the best ones that should be taken forward into the preclinical and clinical trial phases. Again, here going quickly through 3 use cases. So in the first use case, basically what I said before, we got cell we got cell data, and then the challenge was to be able to predict what would be the response after a particular timing point, and then that was actually what we were able to show again and again, that we can actually do those temporal prediction. The second use case, we had pretty much mice and other animal data, and then we were able to predict what would be the response in humans. And then the final one, which is one of also our current use cases, is where in clinical trials we are able to actually predict from data that we get from humans in kind of like a particular timing point to predict a month, 2 months, or 3 months later, or even much later than that, what is going to actually be the immune response and immune coverage at that point. And with that, I would like to thank you all and finish this presentation.